Okay, could somebody confirm that you can hear me?
I can hear you.
Great,
Yes, we can.
need that. It looks like we're a little behind schedule. Today is February 28th, and we're supposed to be starting chapter 13, but we are still in chapter 14. If we're still behind on Thursday, oh, wait a minute. Yeah, we are behind schedule. All right, so let me inform you that you have a quiz this week. The quiz will cover chapter 8 and lab 9, and that is all the material that will be on the quiz. So it's very specific to chapter 8 and lab 9. Any questions about the quiz? If not, let's go ahead and get started with chapter 14 and try and make up some lost ground. So we had talked about this slide here, the development of disease, and mentioned that diseases normally go through 5 stages of disease: the incubation period, the prodromal period, the period of illness, the period of decline, and then the period of convalescence. During all of these periods, you do have the infective agent. You are contagious, although in some periods you're not very contagious. Like at the beginning of the incubation period, you're not really contagious. And then in the period of convalescence, you are contagious, but it's very low. Not all diseases have every period. I mentioned that frequently when people get the flu, or at least often, they may not have a prodromal period. And then some diseases, there is no period of decline or period of convalescence. That is particularly true if the disease kills off the patient. So when we're talking about the spread of an infection, it's important to talk about the reservoirs, and that is a source of the infection, where the infective agent came from before it got into a host and caused disease. There are different reservoirs of infection. They can be living or non-living reservoirs. For human diseases, humans are a major reservoir of disease. So for many human diseases, humans are the main carrier, if not the only carrier, of the infective agent. That's true for AIDS and gonorrhea, where these diseases do not cause disease in other animals. HIV can grow in one of the chimpanzees, one of the species, I don't remember which, but it does not cause disease, or at least AIDS, in the chimpanzee. Gonorrhea and syphilis in particular cannot, well, that's not true. Syphilis can grow in rabbits, but gonorrhea is not, does not grow in other species. So people who are sick can carry a human disease, and they'll be a human reservoir, as well as people who have inapparent infections or latent disease. So somebody who has an asymptomatic disease can spread around the disease, like COVID-19, and they may not even know they're sick. They probably do not, because they have no signs or symptoms of disease, but they have the disease carrying agent. And it can be a serious disease like polio, although COVID-19 is serious, it can kill you. Animals can be other reservoirs. This is particularly true for some diseases like rabies, which this disease does not spread from person to person. So when a person gets infected with rabies, it's a dead end for the virus. The virus will not spread to another host. However, when the rabies gets in an animal, especially a dog or a cat, it can spread from that animal. For rabies, why it spreads so readily from animals but not from humans is the virus does not get into the salivary glands of people, and it does in dogs and cats and most other animals. And so the virus spreads when the animal bites you. Lyme disease does not spread people to people. It spreads tick or a biting insect to people. Some zoonoses may be transmitted to humans. A zoonosis is an animal disease that can be transmitted to humans. So rabies, for example, is an animal disease, but it can be transmitted to humans. So it is a zoonosis. Any question about any of that? All right. And then there can be non-living reservoirs for diseases like botulism, tetanus. These are the normal reservoir of disease. For example, the soil, water, and lettuce give people a large number of human diseases. A large proportion of human diseases come from non-living reservoirs. And that would be obviously botulism and simply an E. coli infection, gastroenteritis, or salmonella from a non-living reservoir. And lettuce was alive before you picked it, but once you pick it, they consider it a non-living reservoir, even though the lettuce isn't quite dead yet. Meaning when we eat it, it's usually green and the cells are alive, but the organism is no longer alive. Any question about any of that? Transmission of disease. There are three principal routes by which an agent of disease can be transmitted from the reservoir of infection to a susceptible host. There's contact transmission, such as direct contact like kissing or a handshake, vehicle transmission, and that would be something like a glass or a spoon, and then vector transmission. That's usually by an animal, like a rabid animal biting a person or a mosquito biting a person and then giving you disease. So vector is by an animal, and then vehicle is by an object. When we're talking about contact transmission, there are three types of contact transmission. Direct, that's person-to-person transmission, and it requires close association between the infected individual and a susceptible host, such as when somebody coughs or sneezes into their hand and then uses their hand to shake their hands with another person, and that causes the infective agent to spread to that person. They're not infected yet. The person who gets it on their hands then has to infect themselves by touching a mucous membrane in the face. And that is actually the number one way that people get colds and flus, that they pick it up on their hands by either a handshake or by touching a light switch, and then they inoculate themselves by touching one of the mucous membranes in the face. There's also indirect transmission, and this is spread from a reservoir to a susceptible host via a non-living object. And we call a non-living object a fomite. For example, when boys are playing baseball and somebody sneezes on their hands and then touches the baseball, the infective agent is spread to the baseball, and then the baseball can spread it to other boys on the team. Any question about any of that? A common fomite spreading diseases in the United States is something like money. Money is touched by a lot of people. And if somebody has a cold, for example, the cold virus can be on the money, and then it spreads with the money. And that could be coins or dollar bills. The third contact transmission, I don't really quiz you on, but it's considered droplet transmission, and that's transmission from an airborne droplet that travels less than 1 meter. So on a cough or a sneeze, the 1 meter mark, right about here, is considered droplet transmission. And that's considered contact transmission. And the reason why I don't talk about that is if it's beyond 1 meter, it's considered airborne transmission. And that's a vehicle transmission. And I just decided that that's a arbitrary distinction, that this 1 meter mark is pretty arbitrary. And it is true that when you sneeze or cough, you have a majority of it within 1 meter, and then it drops off after 1 meter. And so I understand that part, but still that is a subjective boundary, and it depends how violent the sneeze is, for example, how far the droplets will go. And going back to contact transmission, there's direct, and then droplet, which I won't quiz you on, and then indirect, like the baseball. Any question about any of the contact transmissions or transmitting a disease? All right, let's go into vehicle transmission. We have 3 types of vehicle transmission. There's waterborne transmission, where the infective agent gets into water, or contamination of the water by sewage. This doesn't happen very often in first world countries, but in third world countries, it happens more often. And waterborne transmission for the human race is a particularly problem for a source of disease. But like I said, this is more often in third world countries, waterborne transmission. In the United States, generally, we do not get sick from drinking water. The exception would be if your server had a cold or the flu, and then they touched the glass, in which case that's a vehicle transmission, which is a vehicle transmission, but it's not waterborne. And I don't know what you would put this in. It's not waterborne or foodborne. It's vehicle transmission, where the inanimate reservoir transmits it by the cup. Okay. A foodborne transmission is when the infective agent is transmitted by food. That happens particularly with undercooked food, poorly stored food, or unsanitarily handling of the food. Generally, if you go to McDonald's or Burger King, your food should be pretty good. However, if you hear somebody coughing or sneezing, you might not want to eat the food, although the cooking will kill it. If they handle the food after it's cooked, they can transmit the infective agent to the food. And especially the cold would probably survive that. If the food's still hot enough, it probably would kill the flu virus. But the cold would be a little more, it's a little more hardy, the virus. And then the third vehicle transmission is airborne transmission. And like I said, when you cough or sneeze, if it goes beyond 1 meter, it's considered airborne transmission. And it's not contact transmission, it's vehicle transmission. And I just decided I'm not going to quiz you on that because I consider that a fairly arbitrary distinction. Because you're getting it from the cold or the sneeze, I mean the cough or the sneeze, regardless of whether it transmits 1 meter or beyond 1 meter. All right, any question about vehicle transmission? Where it's transmitted by an object, and that can be food, water, or bodily fluids, or utensils like cups and silverware. Okay, there's vector transmission, and that's where the animal carries a disease to a person. The most important group of vectors are the arthropods, especially biting fleas, ticks, and mosquitoes. All of these arthropods bite people, and they can transmit disease. You all know about like mosquitoes transmitting disease when the mosquito bites you, and so that would be a vector transmission. You can transmit the disease by mechanical transmission. So if it's vector-borne, it can be mechanical transmission, and that's passive transport, where the fly passively picks up the disease-causing agent on its feet, and then the feet passively transmit the disease to the food, and then the food is eaten by the human, and then the human gets the disease-causing agent by mechanical transmission. Any question about that? Mechanical transmission is different from biological transmission. Both of them are vector transmission, but in biological transmission, the pathogen actually reproduces inside the vector. So when the malaria is given to the person, it's a biological transmission because the malaria can grow within the mosquito. And that's not passive because the mosquito doesn't passively pick it up and then transmit it to the patient. It's actively transmitted by the bite of the mosquito, but it's biological because the malaria grows within the mosquito. Any question about any of that? We won't talk about malaria any further, but let me mention that malaria does require two hosts to grow. Certain stages grow in the mosquito, and other stages of the malaria parasite grow within the humans. And so malaria requires two species to survive the malaria parasite. All right, let's move on to nosocomial infections. This is a very important topic for you guys. If you're involved in the healthcare industry, when you get out of taking classes, and some of you are probably involved in the healthcare industry already, nosocomial infections is actually probably the most important topic you can learn in microbiology. Although aseptic technique is really important that you learn that too. But nosocomial infections are very important because what they are diseases that are spread to a patient because of their stay in a hospital or hospital-like environment. So the patient would not have picked up the disease-causing organism if they had not come to the hospital or a hospital-like facility. And that can be like a nursing home, a doctor's clinic, something like that. It doesn't have to be exactly a hospital, just a place where somebody who's sick or incapacitated goes. So this is something that was not present in the patient until they were admitted into a hospital-like environment. And nosocomial infections are also called hospital-acquired infections. It's estimated that 5 to 15% of all hospital patients acquire a nosocomial infection each year. And that's 2 million people per year in the United States, or about 20,000 deaths each year from a hospital-acquired infection or a nosocomial infection. It's actually the 8th leading cause of death in the United States. Nosocomial infections occur because of 3 factors in the environment, and that is microorganisms are in the hospital environment. The patient who's in the hospital environment is a compromised host, and then there's a chain of transmission in the hospital-like environment. Let's go back to that slide. So the CDC says that at least 1 out of 20 hospital patients comes down with about a nosocomial infection each year, and they're talking about the United States. Okay. And about 50% of nosocomial infections are preventable. So the patient doesn't have to get them because at least 50% of them can be stopped. It's a major economic cost to the United States. You don't need to know any of these numbers, but I'm just showing them. But like I said, nosocomial infections happen because of 3 factors. The microorganisms are in the hospital environment. Hospitals are a major reservoir of human diseases because when people are sick, they have disease-causing agents that cause them to be sick, and these people go to the hospital. So hospitals are a reservoir of infective agents. Some normal microbiota are opportunistic pathogens, and they can spread as a nosocomial infection. And then worse, some nosocomial pathogens are antibiotic resistant. And so when the doctor prescribes the usual course of antibiotics, it doesn't work. And then if the doctor tries the second antibiotic and it doesn't work, and he says, okay, let's do a antibiotic test, a Kirby-Bayer test to see if there's any antibiotics that work, well, the patient may die by this time from the nosocomial infection. And then in the hospital, because of the presence of antibiotics, we frequently have infective agents which are antibiotic resistant. Or I should just say that they're more likely to be found in the hospital because hospital has a high percentage of patients who are on antibiotics. Much more, say, than most other environments like the grocery store or church services, something like that. The patient going into the hospital is a compromised host, one whose resistance to infection is impaired by either disease, by therapy, or by burns. Broken skin or mucous membranes compromises the host, making them more likely to come down with a nosocomial infection. Burns, injections, ventilators, urinary catheters, the administration of anesthesia, all make a hospital patient more likely to come down with a nosocomial infection. Patients who are in the hospital sometimes have a suppressed immune system that will compromise the host, and that could be caused by drugs, radiation, or burns that weaken the immune system. There's a chain of transmission in the hospital-like environment, which spreads around the nosocomial infection. The principal routes of transmission can be direct contact transmission, such as patient to patient, or hospital staff to patient. This is particularly a problem in special care units, and the hospital knows that. So usually in special care units, they go through rigorous procedures to prevent nosocomial infections from spreading. There's indirect contact transmission, and that can happen via fomite, such as the urinary or IV catheters, contaminated surgical dressing, and in the hospital, catheters and surgical dressing should not be spreading nosocomial infections. But if they become contaminated and then they're used, they will. And then there can be a vehicle airborne transmission, such as when the nosocomial infective agent is spread by the ventilation system. I remember when COVID-19 was first a problem in the United States, they were worried that it would spread in the hospital by the ventilation system. And that's because COVID-19, as well as colds and flus, can be spread by the air. However, COVID-19, fortunately, does not tend to spread that way. It tends to spread much more patient to patient or person to person, whatever you want to call it, that it can spread by the air, but fortunately it does not. And so the ventilation system did not tend to spread COVID-19 in the hospital. And that was a good thing because if you recall, at times of the year, especially when the COVID-19 first hit, and then when it hit the next winter, the hospital was full of COVID-19 patients. There are different steps you can take to control nosocomial infections. There are general procedures that are implemented. You've learned aseptic technique in microbiology, so now always use it whenever you're working in a hospital-like setting. That will prevent the spread of disease. The proper handling of contaminated materials is essential, and you've supposedly learned that in microbiology. If you're handling contaminated material, you should keep that separate. And then when you're done handling that material, you should wash your hands or take off your gloves so that if you got it contaminated on your hands or your gloves, you get rid of it and then you don't spread it around. And then a frequent and thorough washing of your hands has been shown to prevent nosocomial infections. So you should do that. Nowadays, I believe they've told all the hospital employees, you must wash your hands after you touch a patient. Do not pick up a nosocomial infective agent or some other infective agent from one patient and then spread it with your hands to another patient. And the hospital is much better about that. They do wash their hands generally between patients. And then they've educated, of course, the staff to take that step. And lately, the last 10 years or so, they're educating the patients and the visitors. And that's very important to do that because patients and visitors can spread around nosocomial infective agents in a hospital. And they have signs outside of the elevator in the hospital, as well as maybe in the elevator. And then they have hand sanitizer by the elevator or by the patient's door. And you're supposed to use it before you go into the patient's room. And that's to prevent the spread of nosocomial infective agents. There are many other steps you can use to prevent the spread of nosocomial infections. Take a look at the online chapter notes, and they go through some of those steps. Any question about any of that? All right. Let me state again that simply frequent and proper hand washing between patients can stop the spread of nosocomial infections by 50%. So that is something that if you're a hospital employee, you really should do. Otherwise, you will spread nosocomial infective agents. Any question about any of that? All right, that's it for nosocomial infective agents. Let's move on to epidemiology. Epidemiology is a study of where and when disease occurs and how diseases are transmitted in populations. The Center for Disease Control, the CDC, collects and analyzes U.S. epidemiological information. And they publish that in the Morbidity and Mortality Weekly Report, which is usually called the MMWR, which you can find at www.cdc.gov. Notifiable diseases are those diseases that physicians are required by law to report to the local health authorities. So in each state, they have a law, and the physicians must report these to usually their county health offices. What do you call that? The county health department. And then the county sends it to the state. So that would be to, I think, in Olympia. I'm not sure where the state county offices are in Washington. I'm new to this state. In Oregon, it's in Salem. I moved out of that state just recently. And then the state individuals send it to the CDC. So normally the doctors send it to the county offices send it to the state office, and then the state office sends it to the CDC. And then the CDC publishes it in the Morbidity and Mortality Weekly Report each week. And you can see that if you wanted to at www.cdc.gov. Any question about any of that? All right, two more terms you need to know about epidemiology. Morbidity is the incidence of a specific notifiable disease. The morbidity rate is the number of people affected in relation to the total population in a given time period. The mortality is the deaths from a notifiable disease. The mortality rate, the number of deaths from a disease in relation to the population in a given time. I used to know the COVID-19 mortality rate. I do not know the morbidity rate, but my belief is that something like close to 60% of Americans have come down with COVID-19. And so the morbidity rate would be 60% or higher, because my data is probably a year old. And the mortality rate, it's something over 1 million people have died of COVID-19. And I think that's only in the United States. It would be much higher for the mortality rate in the world. Any question about any of that? All right. I only gave you the COVID-19 because those are the two numbers I do know. You can use whatever numbers you want for these, but I just don't know for other diseases. All right, any questions about chapter 14? If not, let's move on to chapter 13. Viruses, viroids, and prions. We're only a half an hour behind schedule. All right, as is normal, I normally show you the major goals of this chapter and give you a rough outline. As always, please note that you should know the terms, okay? Particularly know what a lytic and lysogenic life cycle is. We'll talk about those. What a persistent infection life cycle is, what a latent life cycle is, and we'll talk about all of those. Understand the characteristics, the structure, and the classification of viruses. Understand about viral multiplication. And then lastly, we'll talk about prions, understand what prions are and what viroids are. Any questions about what we're going to be doing? All right, I don't hear anything, so let's move on. You should understand that the goal of a virus is simply to replicate. And how do they do that? A virus will follow this general invasion strategy or replication strategy. They will invade their host, hijack their host cell, and then force it to make virus or viral copies of itself. And then these viruses will then escape from the host and spread the virus. And let's see if I can open that link. I'm going to turn that down because I believe it's a little loud at the start.
In the microscopic world of bacteria, a miniature act of piracy is about to take place. T4 bacteriophages. With their spider-like tail fibers and prism-shaped heads, these viral marauders plan to hijack an unsuspecting bacterium and use it as their own metabolic nursery. This E. coli bacterium has seen its last day. Binding to its surface, the virus injects its precious cargo of genetic material into the cell, commanding it to produce a new batch of T4 bacteriophages. In a final act, the bacterium is forced to self-destruct, releasing hundreds of viruses ready to kill again.
All right, any questions about that? Oh, let's close that down. I don't think we'll continue that, but let's move on. All right, some general characteristic of viruses. Viruses are inert when they're not within their host cell. Viruses display little or no metabolic activity outside of their host cell. They require a living host cell in order to multiply. So viruses are considered an obligate intracellular parasite. That means they have to replicate within their host cell. And without their host cell, the virus is not going to replicate. And without their host cell, they have no or little metabolic activity. They multiply within their living host cell, and they use their host cell's enzymes, nucleic acid, amino acids, and other molecules to reproduce. You should know that viruses have no ATP generating mechanism. How they get ATP is by using the ATP within their host cell. Viruses do contain DNA or RNA as their central most molecule, and we can call that their genome. So it is either DNA or RNA, but it is never both. And this is a little unusual, and that is the genome of viruses can be RNA. You never see that within a cell. Viruses have a protein coat surrounding their genome, and that we call the capsid that surrounds the nucleic acid. Some viruses, but not all viruses, have around the protein coat an envelope, which is not shown in this picture here. And that envelope, we call it has a lipid bilayer. We'll talk a little bit more about that. So some viruses have three, I don't know, structures, and that is a central molecule, which is its genome, which can be DNA or RNA, and then an outer coat that's protein. And this has this section removed so we can see inside the virus. And many viruses are only that. Its genome and then the protein coat, which we call a capsid. But some viruses have an envelope around the protein coat. And that envelope is a largely phospholipid bilayer. But not all viruses have an envelope. Any question about any of that? The host range of a virus is the range of cells that the virus can infect. The host range is determined by specific host attachment sites, such as cell walls or flagella, and these attachment sites have receptors on them that the virus binds to. And there are cellular factors necessary for viral multiplication, such as if a virus requires a specific viral enzyme, not a viral enzyme, a host cell enzyme. If the virus requires that for the virus to replicate, then that's a factor that determines the host range of the virus. Most viruses are very specific, and they infect only the cells of one or a few species. And that's because viruses have ligands on them, or adherins, molecules that bind to receptors, which is where the host has a molecule that the virus binds to. So ligands or adherins are the molecules of the virus that bind to the receptor, which is a molecule of the host that the virus binds to. We're going to talk mainly, well, not mainly, we'll be talking mainly about two viruses, bacteriophages, which are bacterial viruses, and they're called bacteriophages, and I'll try and use that word, but sometimes they're called phages or phages. Both pronunciations are correct. And then the other group of viruses we won't talk about today, but we'll also talk about animal viruses, because they're the ones that make people sick. And this course is, of course, geared to talk about infective agents that cause people to be ill. So that's why we're talking about bacteriophages mainly and animal viruses. Any question about any of that? There are some exceptions where a virus can infect more than one or a few species, and we will talk about some of those exceptions. Okay, but most viruses only infect cells of one or a few species. A prime example is HIV, which only infects the cells of one chimpanzee and human cells. And when we're talking about human cells, there's only a few cell types of humans which become infected by HIV. Obviously, T cells are one cell. I think macrophages and brain cells can be infected, but there's many cells that HIV cannot infect, and that would be like our muscle cells and our skin cells, and there's many other cells. I don't know which cells get infected and which do not, but there's many cell types that are not infected. And I know skin and muscle, probably bone, they're not infected. But brain cells and macrophages, I believe, and certainly T cells can get infected by HIV. All right, let's talk a little bit about viruses and their size. Viruses are generally much smaller than a prokaryotic cell. So right here, we're seeing a portion of a red blood cell, and that's its intended size. That's E. coli, which is a little bit bigger than most bacteria. It's, I don't know, 2/3 to one. If you take 1/2 the size, or maybe 2/3 the size, that would be your average bacterium. So E. coli is a little bigger than your average bacteria. And then let me blow this up. We're seeing some viruses. Some viruses actually approach the size of a prokaryotic cell. Ebola is actually as long as a prokaryotic cell, at least an average prokaryotic cell, but it's not the largest virus. The largest virus is considered megavirus, and we go by its diameter, and its diameter is the size of the smallest prokaryotic cell, and that's about 0.4 micrometers or 400 nanometers. I don't see the size here, but that's what it is. I just know it off the top of my head. And then viruses get smaller than these two largest viruses. The smallest virus you've probably ever heard of is the polio virus, 30 nanometers. But there are some viruses smaller than that. I just don't think, I can't even read this now, just a minute. I don't think many of you have heard of MS2, a bacteriophage, which is 24 nanometers large, one of the smallest viruses, a little smaller than poliovirus, but not by much. So for us, poliovirus would be the smallest virus we've heard of. Any question about any of that? So most viruses range in something from 20 nanometers to 440 nanometers in size, their diameter, their length. They can get a little longer, but they'll be long, and then they are very skinny, the viruses. And so the largest diameter would be 440 nanometers, which is about the size of the smallest prokaryotic cell. And viruses can get a little longer when we only measure their length. And as far as I know, Ebola is the longest virus that we know of. When we're talking about virus structure, we're talking about a virion. A virion is a single mature, complete infectious virus particle. So when a virus gets into a patient and then replicates, what comes out of the patient we call virions. Any question about any of that? All right, in the viral structure, we have nucleic acid, which will be the genome of the virus. And the genome of virus can be DNA or RNA, but it is never both. The genome can be double-stranded or single-stranded. And when we see a double-stranded DNA virus, that's nothing unusual. Cells have double-stranded DNA for their genome, and so do some viruses. Some viruses have single-stranded DNA for their viral genome, and that's not unheard of in cells, because when the DNA is replicating or when the DNA is being transcribed, the DNA will be single-stranded. Viruses can have a genome which is double-stranded RNA. That is unheard of in cells. Our cells do not have double-stranded RNA, unless, of course, the cell is infected with a virus that has double-stranded RNA. But viral genomes can be double-stranded RNA. Viral genomes can also be single-stranded RNA. When we have a single-strand genome in a virus, we need to specify whether that genome is the positive strand or the negative strand. So we have two single-strand RNA genomes, positive single-strand RNA and negative single-strand RNA. The positive single-strand RNA, this RNA can be read by the ribosomes. So they would have like AUG, for example. The negative single-strand RNA would be the complement to the positive strand of RNA. The ribosome cannot read the negative single-strand RNA genome. And it would not have AUG, it would have UAC, for example, the complement to AUG. Any question about any of that? All right. When we're talking about viruses, their genome can be linear or their genome can be circular. If you remember, eukaryotes have linear genomes, a linear chromosome, and then prokaryotes, at least bacteria, have circular chromosomes, circular DNA molecule. And viruses, depending on the species, will have one or the other. Some viruses, like influenza, has their genome in several segments. And that should be familiar to you because humans have their genome in several segments. In humans, we call these genome segments chromosomes. Okay, and viruses like the influenza virus, we don't call them chromosomes. Any question about any of that? All right. As I stated, when we're talking about the viral structure, the virus can have a central molecule, which is the innermost molecule, which is its genome, and it can be RNA or DNA. And surrounding that genome will be the capsid, which is a protein coat. The capsid, let me blow this up. The capsid is composed of subunits that we call capsomeres. So this virus here will have a capsomere right there, and another one right there, and another one right there. And it looks like this virus has only one protein or capsomere that makes up its capsid, the protein coat. That's not true of all viruses, where they may have more than one protein to make up their capsid. Any question about that? All right. If we have a virus that is only its genome and its capsid protein coat, we call this virus that has no envelope, we call it a naked virus. So this is a naked virus shown right here. If the virus has an envelope, we call it an enveloped virus. And that envelope is composed mainly of a lipid bilayer. It does have proteins and carbohydrates in it. Oftentimes, if the virus has an envelope, it will have spikes on it. And these are usually glycoprotein spikes shown here. And the spikes will be the outermost portion of the virus. And so these spikes will be the antigens projecting from the envelope surface. An antigen is what our host immune system recognizes as foreign, and it usually recognizes molecules that are the furthest most out, or the outer molecules of the infective agent. So the spikes are the outermost molecule on this virus, and so the spikes will be the major antigens on this virus. The spikes serve as attachment points for binding to the host cell, and usually the binding points on the virus are the outermost portions of the virus. So the spikes for the virus, it uses the spikes to attach to its host cell receptors, and then the spikes are recognized by the host as, are recognized by the host as antigens to use or recognize this as belonging to not the host, meaning the host immune system will respond to these antigens. Okay, for this naked virus, what part of this molecule is going to bind to the host receptors?
The spikes?
This molecule has no spikes. I'm talking about the naked virus, not this one here. Come on, I've already told you.
Capsid?
It's the
Capsid?
outermost molecules of the virus, and so it'll be the capsid proteins will bind to the, come on, mouse. The capsid proteins will bind to the receptors of the host cell. And so it's always the outermost molecule of the virus, which is the portion of the virus that binds to the host cell receptor. And we have a name for that. Like I said, they're either called ligands or adherens. I don't think I test you on that, so you don't need to know that name. I'm not even giving it to you in this lecture, so like I said, you don't need to know that name. All right, when we're talking about the viral structure, it's important to talk about the general viral morphology, and that is the shape that the viruses are in. We classify viruses into 3 different shapes based upon their capsid structure. There's the helical virus, shown here, and that's clearly a helix. I look at that, what is that, Ebola virus, and they say it's helical, but I sure don't see it. If someone can see it, you can point it out to me. And then there's the polyhedral virus, which has many-sided virus. It sort of looks like a fancy dice. If you've ever had a multi-sided dice, polyhedron. And then there's a third general morphology of viruses, and it's called a complex virus. A complex virus can take on essentially any shape that's not helical or polyhedral. The bacteriophage T4 actually looks like the lunar lander, if you remember what that was from the Apollo missions. Here's another complex virus. I think that's rabies, but I can't read it. So anyways, it's just a complex virus, and it looks very different from this complex virus. This is a complex virus. It's not showing it. It's showing you more the envelope, which let's blow that up. It is a complex virus, but it's showing you the envelope. And there we see the envelope around the capsid right there, which oh, here it is. Wow, that's two capsids inside the envelope. There are some viruses like that. So there's the capsid right there. And then there's the nucleic acid. All right, let's talk about bacteriophage T4. It belongs to the T-even family of viruses, a complex virus in this shape. The whole family takes on this shape. T4 we're going to talk about specifically because it's the virus that has been studied more than any other virus. And the reason why T4 has been studied more than any other virus is simply because virus T4 infects E. coli. And E. coli is the cell that microbiologists frequently have in their lab. And so when people were first starting to study viruses, they keyed in on T4 because it's a virus that infects E. coli. And we know more about the virus T4 than any other virus in the entire world. Any question about any of that? When we have an infection that's a viral infection, the host immune system will respond. Meaning a virus infects the host, that will stimulate the host to produce antibodies. The antibodies will bind to and recognize viral surface proteins. And these may be glycoproteins, and we call what the antibodies bind to antigens. Antigens are what the host immune system responds to. And why the host is doing this is the host can neutralize the virus by binding to it with antibodies. And then we won't talk about it, but the antibodies can help clear the virus from the host. Some viral proteins can change or mutate frequently. And then that will mean that the host will have to make new antibodies to then bind to the changed or mutated virus. Different viral strains can therefore infect the same individual more than once. And an example of this is, of course, COVID-19, where the virus surface proteins change, and therefore you can become infected by it more than once. And you might be infected by it more than once also because the host immune response fades over time. But the main reason is that the virus is changing. And so if you become immune to an original COVID-19, later in time, there will be a new COVID-19, which will get through your immune system, meaning your immune system will not recognize it or recognize it well. And so you can get another infection. A better example of a virus that changes or mutates frequently is the influenza virus. And this virus changes or mutates more often than any other virus that I know. And that's why when you get a vaccine to the flu, it will only be good for at most about 3 years. After 3 years, you need another vaccine. And that's simply because the flu virus changes that rapidly. COVID-19 does change, but not quite as rapidly as the flu. It does change much more rapidly, though, than most other viruses. Many viruses do not change that often or that frequently. They do change, but very slowly. For example, the what's that? The polio virus. They made the vaccine for the polio virus in the 1950s. I think one vaccine was made in 55 and the other one was made in either 57 or 58. Those vaccines are still good today. That's over 50 years. I think that's actually about 70 years or close to 70 years that the vaccine has still worked. And so for the polio virus, it's not changing very rapidly. And that's a good thing because then we don't need to make the polio vaccine again. But with the flu virus, as you know, they make a new flu virus or vaccine, sorry, they make a new flu vaccine every year. And they would have to do it every 3 years because it changes so rapidly that after 3 years, the vaccine is no longer any good. Any question about any of that? All right, let's move on to viral taxonomy. Viruses are grouped into families. They can be grouped into, let me see now, King Peter's son came over from, they can be grouped into orders, but higher than orders, viruses are not usually grouped in, and they're not grouped in anything higher than that, except they will be grouped into the kingdom that they infect. So we do group them into like animal viruses, they only infect animal cells, but we don't group them into, let me see if I can remember this now. King Peter's, we don't group them into phylums, orders. King Peter's son came, whatever it is, I don't remember. We don't group them into much higher than family. We do group some of them into orders, but generally viruses are grouped into families, and then we group them into genus, and then we group them into viral species, and then we group a viral species into subspecies. A subspecies for the human race would be the different races, like Caucasian or a Negroid, or what do you call Mongolian or Oriental? Okay, that would be what the subspecies are. So viruses are grouped into families based on the type of nucleic acid they have, what their replication strategy is, and what their morphology is. The family names always end in viridae. Like Pox viridae or the Pox family of viruses, and it's Pox viridae for the family name. The genus name ends in virus. So for the Pox genus, it would be Pox virus. The viral species is a group of viruses sharing the same genetic information and ecological niche. So that for the Pox family of viruses, we would talk about smallpox being the species, one species of the Pox family. Okay, Pox viridae. Any question about any of that? Virus species are given common English names. We have no scientific Latin naming system. What happened was the first virologists were all English speaking, and they started naming viruses with common English names. So the polio virus is simply called the polio virus. The influenza virus is simply called influenza virus. And that is its species name, the common English name. And surprisingly, when virologists came about in other countries that spoke different languages, they adopted using the common English names. Any question about that? All right. And now subspecies are designated by a number like herpes simplex. There's type 1 and type 2. These are subspecies of the herpes simplex species of virus. The subspecies are a very important distinction in viruses. I'm sure you know that herpes simplex 1 and herpes simplex 2 essentially give different viral diseases, right? So the subspecies is very important in viruses, but let me point out exactly how different the subspecies can be. We have poliovirus 1 and poliovirus 2, which generally aren't named that. They're simply called the poliovirus, which causes the disease polio, which is a very serious disease. And then we have the Sabin vaccine, which is a subspecies of the poliovirus, and we use it as a vaccine to prevent getting the disease polio. So that tells you how different the subspecies are, at least in the respect of causing disease in humans. That the poliovirus, one of the subspecies causes the polio disease, the other subspecies we use as a polio vaccine to prevent getting the polio virus. Any question about any of that? All right, are you happy that the viral taxonomy, we use common English names to name and taxify, or whatever you call that, characterize viruses? Does that make you happy? You'd rather know Latin names for viruses? All right, let's go to the example of the virus taxonomy, talking about the family herpes virus. The family name is herpes viridae. The genus name is herpes simplex virus. The common name is human herpes virus, such as human herpes virus or human herpes simplex. That would be the species. The subspecies would be human herpes simplex type 1 and human herpes simplex type 2. We generally, the scientific name is not taken by this virus, where it's called human herpes virus. It's more often called herpes simplex virus. Okay. Most people have used the older name human simplex virus, and except maybe in a textbook, you probably won't hear the term human herpes virus. It's a name that just never took. Any questions about any of that? If there are no questions about that, I think, let's see how we're doing here. No, I better go on. We're a little behind schedule. You can grow viruses in the lab. If you're growing bacterial viruses, their name is called the bacteriophage, and I've actually grown them, where you grow bacteria on a plate, and what you have, let me see if I can blow this up, is a lawn of bacteria. That's why the plate is kind of whitish in this area, meaning the bacteria is growing everywhere on the plate. And then you put on the plate a solution containing a virus, and then you either spread that or dilute it, whatever, you put that solution everywhere, and then you incubate it. And if there's a virus there touching a bacteria, the virus will infect the bacteria and then use the host bacteria cell to replicate, making multiple copies of itself, lysing the bacteria. And then where that virus goes? It goes to nearby bacteria. It will infect those cells, lyse those cells, release more viruses, which will go to nearby viruses. And once you incubate this, you will eventually get plaques on the plate. And for each plaque, that is where a virus or a viral particle started, meaning it could be a clump of viruses. And then you can count the plaques and determine how many viruses you have in solution, depending on what your dilution was. Okay? I've actually grown viruses this way. And like I said, the bacteriophages actually grow in the bacteria and then form the plaque. What happened here is, let me blow that up a little further so you can see it. It looks like this one was maybe 3 plaques that fused together. That one is probably 1 plaque, and certainly that one is 1 plaque. And it's larger than this plaque, probably because initially a virus touched the bacteria initially, and so the virus replicated right away. Whereas this plaque and that plaque are smaller than these 2 plaques, probably because initially the virus was not touching a bacteria, and the virus had to wait until the bacteria grew. And then when the bacteria grew enough, eventually one of the bacteria would touch the virus, and then the virus could then begin replicating. Why that plaque and that plaque are smaller than this plaque and that plaque. Any question about any of that? So that's growing up bacteriophages in the lab or viruses in the lab. When we're talking about viral multiplication, you should realize that the viral genome only contains a few genes, such as the capsid proteins, and any other genes needed by the virus to replicate itself. How the virus replicates is the virus will hijack its host cell, take control of the host cell's metabolic machinery, use the host ribosomes, tRNA, amino acids, ATP, and some of the host enzymes to replicate the virus. And then the basic viral multiplication is similar for all viruses. And viruses that infect a bacteria called bacteriophages follow 2 mechanisms to replicate: the lytic life cycle, where the host cell is lysed and killed, or killed and lysed by the replicating virus, and the lysogenic life cycle, where the host cell remains alive, and the bacteriophage and the host cell will live in symbiosis for at least a time. Any questions about viral replication? If not, let's leave it here, and I'll see you at 6:30 for the lab. All right, goodbye.